Chapter 16, Digynians. Part 4, Superfamily Schistosomidia, Biology. Now, when you look at the biology of schistosoma, what you're going to see is there's going to be no intermediate arthropod. There's going to be a lack of something that has to come between us and what's going on. The adults are going to be living in our abdominal veins. Okay, depending upon the species, it's going to be, well, it will determine whether or not they want to be, veins are, veins associated with the large intestine, the bladder, or whatnot. Turns out Hemobotany has a tropism. It prefers veins that are draining the musculature of the urinary bladder plexus. Okay, got to remember the bladder is a highly muscularized organ, so it's going to have lots of blood flowing in, lots of blood flowing out. So it's in those veins Hemobotany seems to want to reside. Mansonii wants to go in the large intestine. Okay, wants the portal veins of the large intestine. Remember, the portal vein is the vein that is draining the blood, the highly nutrient-dense blood from the large intestine will be delivering it to the liver. And then Japonicum likes the veins of the small intestine. Again, doing the same thing as Mansonii. The adult females, okay, are very rarely going to be found in the blood on their own. It's one of these things. The male and the female will approach their tropic tissues, and they won't lodge themselves in there. They won't house themselves in there until after they come in contact with their opposite number. Okay, a female has to get into the gynecophoral canal of a male. Then and only then will they work themselves together up into their veins of choice. At which point in time, as they're doing this, working their way up, there's going to be the exchange of sperm. There's going to be, fusion of the two gametes, zygote form, egg formed, and once they're, by the time they're where they need to be, the female is going to be producing eggs. Eggs will be passing out. Now, what's interesting is, depending upon the species, there's going to be distinct shapes. The overall shape, this distorted oval shape, that's not anything we can use diagnostic. What we can use is the positioning of where these little nubs are, those little spines. Where are they coming out? Now, the eggs are going to float in the bloodstream for a very short time, under perfect conditions. They're a very short time because they're going to adhere to the wall of the capillary or the vein that they happen to be in, and they're going to somehow enter into the tissues. They are going to pass through the epithelial wall, get below the epithelium layer into the supporting layers, the musculature, the other, epithelial cells that may there through the mucosa. How they do this, we don't know. Okay, how are they physically doing this? We have no idea. We assume it has something to do with the proteins that make up those spines. We also think it may also be partially due to some form of immune response. The initial entry into, don't know. Maybe it's spine derived, don't know. But the passage through the underlying tissues, that may be host cell, host tissue driven. Um, the cells, the immune system is going to recognize this as a foreign invading body and are going to try to move it away. As the egg is moving, as it's passing through the tissues, a granuloma will form. Okay, granulomas are going to be layers of eosinophils and macrophages that are just going to layer on top of the egg, trying to wall it off. So whatever this invading organism is, it's now surrounded by layers of host immune cells that are shepherding it through, trying to keep it walled off. Once this granuloma passes away all the way through the tissues and then enters into the lumen of either the bladder or the gastrointestinal tract, the eosinophils and macrophages will disperse. The egg is now released on its own. And you look here in this picture, right here. We see under microscopes, we see under the electron microscope, the picture here, that the eggs, once they start penetrating, once they associate and start penetrating in, the immune cells start to gather around. So, again, don't know how the initial penetration, what's causing it, but we do know the movement through seems to be correlates with a strong immune response. Correlates, don't know if that's causation. Here's the problem though. It's an egg. Eggs don't know direction. Eggs don't make decisions. Eggs just adhere and then transverse through the tissues. What happens if they floated in the bloodstream just a little too long? Or what happens if there's a lot of eggs released all at once? Well, the first one, you can get quite a few eggs that are leaving out in the same area. You can start damaging all those immune cells. That strong immune reaction in that one given area is going to start altering the architecture. I know I use that term a lot, but think about it. Architecture, it's how buildings, how things are made. Well, the tissues, the different layers of the epithelium, the endothelium, the mucosa, the muscle, the fat layers, all of that have a certain way they've been laid out. You get a strong immune response going on there, and remember, the immune response can be a little indiscriminate at times. You're going to get damage to those different layers. You're going to get parts, proteins, things like that that are supposed to be part of a layer are going to get damaged and removed and maybe not replaced. If they're replaced, it's not going to be like with like, it's going to be with something different. So this fibrous tissues, all the collagen and things that are supposed to be wrapping around, holding tissues together to form organs and all of that, that collagen gets broken down, it's going to be replaced by fibronectin, it's going to be replaced by other fibrous tissue, fibrous things that, well, for all intents and purposes, it's going to be like an internal scar. That's going to change how that tissue can or cannot function. And then that first one I mentioned, what happens if they stay in the bloodstream just a little too long? They don't exit out immediately. Well, they can get carried within minutes everywhere in the body. And then they would adhere to the walls of the capillary beds, and they start exiting out wherever they may be. The heart, the spine, the brain, the lungs. Not good, Batman. You start to get granulomas forming in your brain, in your heart, in your lungs that will impair, if not irreversibly damage, certain organs. Now, how the eggs pop open and things come out, we don't know. Okay, the myristidium are just going to exit out of the egg. The egg's going to open. It's got its own version of an operculum. It's going to open up. Out comes the myristidium. That's going to be covered in cilia. So they're going to be highly motile. They're positive phototrophic. So that means they have nerve endings, okay, that recognize light, dark. So they will orient this way. So that way they're not swimming in weird directions. As long as they're swimming parallel to the surface of the water, there's a good chance they're going to run into something that they need. They're looking for specific host snails. You see here, Myristidium is being released, coming out of the egg. It is fully formed. It is ready to go. Myristidium enters into a snail. Within minutes, hours, it sheds its epithelium. Okay, it sheds its current version of a tegument. Goes through a metamorphosis, literally. Yes, I use that term a lot in the live lectures, but it is almost on the same scale of caterpillar to butterfly metamorphosis. It's a complete rearrangement of organs and overall phenotype morphology. So the Myristidium becomes a sporocyst. Now, takes a couple of weeks for this sporocyst to become what we think of as a mother cyst. All right. Remember, the Myristidium has stem cells, germinal tissues inside of it. When it becomes a sporocyst, it still has those stem cells, still has those germinal tissues. It's just now those stem cells and germinal tissues are going to become other sporocysts. So you have one sporocyst, and within it, many sporocysts that are slowly maturing. And it'll slowly give birth to these. They will pass out. Live, living things will pass out of it. After a couple of rounds of this, the sporocysts are going to start giving birth to cercariae. Okay, the cercariae which are going to leave the snail. The snail leave the snail, and they're going to hunt. They're going to hunt for us. At the anterior end, what they call the head organ. The head organ has two things going for it: a penetration gland and a ventral sucker. The ventral sucker allows for adherence to us. The penetration gland is going to release digestive enzymes. They're going to break down our epidermis, allow for that cercariae to start penetrating through the epidermis to the dermis and get in. It's interesting, the hunting pattern we see when we visualize, we watch, and we track Schistosoma. They do this up and down swimming pattern as they're going up and down. So that way they're going up and down through the water columns. It's almost a search pattern where they're looking for different organic compounds. Turns out, as you're sitting there watching this video, you are emanating, giving off organic compounds into the air. Well, same thing if you're standing in water, you're giving those off. Now, by going up and down through the different layers of the water column, there's a chance that they will come into contact with those organic compounds, and then they will change and they will start swimming, going up the concentration gradient. One of them that we know, organic compounds we know, is arginine. Okay, they like arginine. Arginine is one of those organic compounds they will respond to. When they come into contact with us, they actually even give off their own arginine, increasing the concentration, in the water. Attract more schistosoma. Increases the chance of a male and a female being in the same definitive host. Takes about 30 minutes to penetrate from our epidermis into our dermis to get into the underlying layers. And then it's going to take about 24 hours to get into the circulatory system because it's got to work its way through the underlying dermal layers, the fatty layers, the muscular layers to get to capillary beds that are large enough, have large enough diameter so that they can enter in. It's all going to be based on that penetration or those head glands. They're going to allow them to move through in between the tissues. Once they get in, they start moving around. They start flowing through the blood. Quite a few of them are going to get expelled in the lungs. Okay? For some reason, they get into lungs. You cough them up. You don't know it. Because remember, these are still microscopic multicellular organisms. You cough them up in the phlegm. They go down your stomach. You digest them away like you would a Big Mac. There you go. Some of them are going to pass through the liver and the mesentery. The mesentery, remember, that's all the stuff holding your innards, your liver and your gastrointestinal tract and your spleen, holding all that stuff in place. Many of them, it's once in the liver, they start to pair up. It's interesting. Males can survive for a while. The females can't. Unpaired males do have a bit of a survival. Unpaired females can't because they have evolved to require that groove, that gynecophoral canal. That gynecophoral canal, the musculature of that aids in the movement of food, aids in bringing in food particles into their oral opening and into their digestive tract and moving stuff through. So as the male is doing stuff, its muscles are moving and it's moving. It's kind of pulling and aiding the female in feeding. An unpaired female has issues, cannot mature because it is on the edge of starvation. It's having trouble getting in enough food particles, thus keeping up its own nutrition. We also see that our immune response is required. Certain cells, certain cytokines, things like that, at certain stages in the fluke development are required. Interfering with those signals, those immune responses, will slow down maturation. Doesn't completely inhibit it, but it slows down the maturation process.